Hi, I'm Beth Moorfield. I'm an editor at Nature Structural and Molecular Biology, and I'm speaking with Jay Dunlap from the Geisel School of Medicine at Dartmouth, and who spoke on session 3 on Wednesday. So, the talk has been recorded, so anybody who would like to go and see it, who have missed that session, can actually access it. And I just had one thing I really appreciated about this symposium is it really underscores the wide variety of organisms in which biological timekeeping is studied, and in your case, Neurospora, right? So I wanted to ask what initially brought you to the field of circadian biology.
So I was a graduate student with Woody Hastings at Harvard. I started in 1974, and Woody worked on Goniolax, which is a system that's fallen out of favor.
Yeah.
But it had a very easy to assay rhythm, and a lot of fundamental properties came out of Woody's work. He discovered temperature compensation, for instance. It was a unicell, so he was one of the first to show that rhythms were a cell autonomous phenomenon, which was highly controversial at the time. Many people who worked on animals thought it was going to be a cell-cell network thing, which it turned out not to be. So it was a very useful system for framing questions. And by the time I finished graduate work, and it was also obviously a problem of importance that no one had any idea how to attack at the molecular level. And recombinant DNA technologies emerged during that period. And so it was plain that the right way to go was to use genetics to identify genes and then use molecular genetics to clone them. And when I finished graduate school, Neurospora could be transformed and flies could not. And those were the two systems where there were mutations available. So I went to work on Neurospora, where Freak had been identified, but obviously not cloned. And so that was 1979. I wrote a fellowship to clone clock genes. Yeah.
Wow, nice. Well, it certainly covered a lot of ground since then. It's really strong organism, especially for these kinds of studies. So, and I think it's especially in this area, people very much appreciate Neurospora. And in your case with your talk, right, you really highlighted, like 2 different aspects of casein kinase 2, casein kinase 1, in the regulation of oscillations, right? I mean, you had described that, of course, there's the negative feedback loop that exists in Neurospora, like it underlies all clock regulation. But in your case, there were 2 aspects of casein kinase 1 regulation and function that were essential, one in the nucleus, one in the cytoplasm, and that you described. And I wanted to know if you could just briefly describe them, because you had some very good questions that you got after your talk in each one of those areas.
So the principal circadian function for CK1 would be in the nucleus. So it forms a complex with Freak and FRH, and it's carried into the nucleus, and that's where it phosphorylates the white collar complex and turns off its activity. So I'm drawing a blank on the cytoplasmic.
Sorry, I was thinking in terms of both Freak and CK1 mRNA regulation, right, by HER2. Did I get that wrong in your talk?
No, you got that correct. So the regulation is apparently happening in the cytoplasm. The regulation of CK1, very likely. So the observation, stepping back, Jerry Feldman Feldman was actually the first to think of making clock mutants, or at least the first to begin this. And about 6 months after Jerry got to Caltech, Ron Knapp came as an early graduate student and spoke with Jerry, and also picked up the idea of making clock mutants, although he published them first. So one of Feldman's mutants that he developed during the late 70s, early 80s was a mutation called period 2, PERD2, PRD2, which is not the same as PER2 in flies.
Right.
And PERD2 is the last of those mutants, many mutants that had not been cloned. So we finally cloned it, and actually we cloned it about 8 years ago, but we couldn't make any sense of what it did. It was mostly disordered, but we finally figured out that this is a very clever postdoc in the lab, Tina Kelleher, saw that it looked like an RNA binding protein. So she showed that it bound to RNA, and did RNA-seq and found that CK1 levels were the thing that was mostly affected. So PERD2 is an RNA binding protein. It binds to CK1 message and stabilizes it. So absent PERD2, there's less CK1, and that results in a long period length. So all of that fits quite well. And the interesting and intersectional part of this was, while we were studying this, a grad student in the lab, Brad Bartholomew, had developed a single molecule fish and could see where CK1 was located, individual transcripts in the cytoplasm, which I think is just a miracle. You can actually do this. And so he asked some straightforward questions. Is it all over the place, or is it concentrated? And it turns out it's concentrated near the nucleus. And this is looking at freak transcripts. Right. And so we had this technology, and turns out that PERD2, when you lose PERD2, you lose this clustering of freak transcripts near the nucleus. And meanwhile, Tina had showed that PERD2 binds to CK1. So the inference will be that they are all bound by this RNA binding protein in a perinuclear space, and that in some way, and here the dots haven't been connected, but the presumption is that this RNA binding protein is in some way facilitating the formation of the negative arm complexes before they move into the nucleus. And when you don't have PERD2, or you don't have these whatever it is that PERD2 does, it takes longer to form the complexes, and so you have a long period length.
Right. No, it was really nice. You could see the image. I guess we should remind people who are listening to this that Neurospora is multinucleate, and so you could really see on the images you had. Yeah, you could see the clustering around.
More like a muscle cell than a fibroblast, right?
Right, but also when you were looking, you had the live cell imaging of this, which was that was really, and it really looked like you even pointed out that it really looked like PERD2 was being kind of dragged along as the nuclei were moving. So, do you have any idea like what might be tethering it? Do you have any idea of like is it anywhere near the nuclear pore complex? Are there any nuclear envelope proteins that you think are helping to keep it associated?
Not so far as we know. We've done pull downs and nothing turns up, nothing suggestive. So I guess I showed, but I haven't mentioned since we're talking that in vitro PERD2 will form these non-membrane bound liquid phase separated blobs, for one of the better words. And this is something that's been trending over the last couple of years. Yeah. And so it's not hard to form blobs in vitro with many proteins. The hard part, and this is both the strength and weakness of the field, the hard part is to show that they actually are something real in vivo, and very few, if any, strong studies have actually connected blob formation with a biological phenomenon, where you can show that something that affects blob formation actually has a phenotype.
Right, it's very much emerging now.
Yeah. So this to us was the interesting part of the PERD2 story, that you can you can make blobs in vitro with PERD2, and freak RNA will go into the blobs. And they're salt and concentration dependent, as they should be, and all those sorts of things. But you can actually, using the live cell imaging, you can see that PERD2 looks like it's a semi-viscous thing within the cell, and it when nuclei move, it tends to move with them.
Yeah, that was very, very apparent on the imaging. It was really nice to see. But I mean, have you been able to isolate them? Do you have any idea whether or not there are any other components that are in the condensates that you're seeing?
We don't know at this point whether there are other components. We would suppose that CK1 is there, but we haven't shown that yet.
Right.
And we don't know what other components might be there.
Yeah, and you're saying that you see two different populations, though. So there's like the perinuclear population, right? And then you're saying that there's separate ones that remain in the cytoplasm not associated with nuclei. Did I understand that correctly?
We haven't shown that. So
Okay.
I would suppose that that's the case, but yeah.
And I wanted to know whether or not they were stimulus responsive. Do you see them either form or not form, right, in response to light or temperature or something that you would expect to be part of the, I mean, to influence the circadian cycle?
None of the above. PERD2 is, I think it's weakly rhythmic in its expression, but not strongly. So that means it's always there. And what is rhythmic are the things to which it binds, in particular freak. And so it may be doing other things with other proteins at other times of day. And so this is just a window on its function.
Right. No, it was really, really nice. I did have I had one more question just in mind, and I just lost it. Right. But yeah, I don't know if you've dissected, its interactions more, or maybe that's ongoing, or you already said when it was first cloned that you recognized that it looked like an RNA binding protein. So I just thought it had normal like RNA recognition motifs that were. Yeah,
it has. Has two different RNA recognition motifs, like the name RM3, R3M, and an SUZ, and it's the SUZ domain, which is less commonly associated with RNA binding, but that's the domain that is required for function. That is, we've done domain knockouts looking to see what rescues, and it's the SUZ domain that is required.
And this is also required, obviously, then for formation of the condensates that you see.
Don't know that.
Yeah.
Well, in fact, it's probably not. The condensates. The dogma is that the condensates form with the intrinsically disordered domains of these proteins.
Right. Yeah. So I guess some are RNA dependent and are dependent on RNA. Or that's why I was also wondering what else might also be in there, because yeah, the function of these, it's just really I just very much appreciated in your talk that you actually could see a function, clear function that was associated with these. But I wanted to know, like, so you're going to have a lot more emerging in this area, I'm sure. And I just wanted to know, like, kind of like backing up to the other aspects of the conference, what areas that you think, or questions that were presented that you are most looking forward to seeing, resolved or further investigated.
It was a conference that focused a lot more on sort of higher level function, and a lot less on mechanism, than I usually follow. So a lot of talks and a lot of information on things that I don't normally pay as close attention to.
Yeah.
I thought one of the areas that I think is going to grow a lot will be the cell biology. So there's also Occam Cromer's talk, which
Yeah.
was really quite beautiful. And
Yeah, right after yours, I think, right?
and also Mick Hastings.
Mhm.
Although he didn't dwell on it, I believe they're doing a lot of work with Andrew Loudon's lab.
Mhm.
And in fields where cell biology has not been pursued a lot, when it comes into focus, not to mix metaphors here, it's really impactful. And I think that'll be true in circadian biology. And when people actually see the proteins moving. And there's always an interface between biochemistry and cell biology that's not immediately, that is, there are paradoxes that will emerge. And we saw some of that in Occam Cromer's talk. That, for aficionados, for instance, he showed that there was a lot of Cry1, there appeared to be a lot of Cry1 in the nucleus. And yet Chuck Weitz has shown that you can clear all of the Cry1 out of the nucleus by immunoprecipitating with PER. And those two, the simple interpretation of both of those observations can't both be right. You can't have tons of Cry in the nucleus and yet have it all immunoprecipitatable from nuclear extracts.
Right.
And so it's, but of course, in the nucleus, in Neurospora, the nucleus is about 5% of the cell volume. So if you, and I think, maybe a little more than that in mammalian cells, but nuclei are not most of the cell. They're a small part of the cell. So if you concentrate all of the protein in a small part of the cell, it looks like there's a whole lot there, but maybe there isn't compared to.
Right. And I think he didn't it's also like he saw like multiple complexes, right? Or right? I mean, they were different populations, right?
Yeah. So there are going to be some interesting things to resolve, and the cell biology is going to point in ways that people don't know yet. So that'll be an interesting thing to follow, I think.
Yeah, well, I mean, again, the recently emerged technology to be able to do live cell imaging is fantastic. It's really nice to be able to really bridge that gap in this idea.
Yeah, and you can do things now that you couldn't do even 5 years ago. The first time we tried to, that is, the technology has improved. The first time we tagged proteins was about a decade ago, and we can see those constructs now, but we couldn't see them then. The self-same constructs in the same cells were invisible at that time.
And can you do multi-tag labeling in Neurospora? You can, okay.
Yeah. So you're only limited by the optics, how many different tags and filters and lasers you have. So you can see about up to 4 at this point.
Wow. Are you pursuing those studies as well?
Oh yeah.
That's going to be really neat. So, so how big is your group?
Smaller at this point. There are 5 students, 3 postdocs, a technician, and Jennifer and myself.
And are most interested? I mean, are more students now, do you find, interested more in the cell biology aspects of this?
Not necessarily. It's something we've been pursuing for a long time, and it's come to fruition now. I'd say there's this terrific graduate student, Brad Bartholomew, really really leaned on this. But I'd say it's the usual mix of some people are interested in some things, some people are interested in other things. So cell biology versus molecular biology versus genetics, and 2 people working on adipocytes, a separate type. Yeah.
Oh, well, I have a feeling that after your talk, you're probably going to see an even bigger spike of interest in pursuing these questions in your lab. But it was really, really good to see you again. But I want to thank you for taking time to speak with me. And I hope you recover. Are you back? Are you back now? Have you returned?
The lab is mostly back. I'm still working mostly remotely.
Okay.
So we're on the cusp.
Okay. Almost fully open then, right? Okay. Yeah, well, now we're getting vaccinated. I hope, I hope we all will be circulating. And the next time that there's another meeting in this area, that will actually all be face to face now. So
The Gluten conference will be face to face in October.
okay. Okay, well, all right. So once we get to the fall, maybe normal life again. Well, again, thanks very much. And
Nice to meet you.
yeah, I wish you well.
Okay.